And today is the third lecture, and we basically finished the overview part of it, and now we're really getting into the details of the data analysis, how we're going to do that, which means from today, the lectures will become boring. And so just to bear with that, keep that in mind, okay? So a little bit refreshing your memory about what we have talked about last time. And the major topic is these sequencers are fast, but what we can do with it. The major applications including many different domains that include the sequence of DNA, which include the de novo sequencing, which means that at the time we do not have a reference genome. We just grab a species, we sequence it. The second one for most of the applications in medical school here is the second one, which is reference-based resequencing. Normally, this is for human, mouse, rat, Drosophila, C. elegans, and more those species that we already have the reference genome. So when we do it, our goal is not to see what the genome of this species is, rather is how different my DNA is compared to the reference genome. So now we are coming into the game of how to identify the SNPs, how to identify the copy number variations, insertion deletions, and so on. The third application is for metagenomics, which means identify who is there in a mixture of microbiome species. It turns out to be very important for environment and also for human disease, because in human bodies there are a lot of microbiome species. And NIH also launched this human microbiome project, which is studying the impact of those species on the human disease. So in addition to sequence the DNA, we can also sequence the RNA molecules. That will include the messenger RNAs, the large non-coding RNAs, and the microRNAs. And we also can use this to study the protein DNA or protein RNA interactions. That will include the ChIP-seq, and CLIP-seq is for the RNA version of it. And combining all this together, we can study epigenomics, and that includes the DNA methylation, histone modification, nucleosome positioning, chromosome looping, and so on. So throughout this course, we're talking about most of these individual topics. So the second part that we discussed last time was the public resources that is available for the next generation sequencing data. So we went through the 1,000 Genomes project. And the goal for this project is to identify the genetic variants across major populations. Again, that human genome project helps us define the DNA sequence of a human species, but 1,000 Genomes is trying to figure out how different we are. The GTEx project is trying to provide a reference RNA level of a signal for different tissues. So they have done over how many? 12,000 of those individual samples across hundreds of individuals and across 50-ish different type of tissues. And ENCODE project, the goal here is to annotate the DNA. So we got the letters of ACGTs, but we want to know which part encode for gene, which is a promoter, and what are the enhancers, and different levels, what the signals really means. And there is a similar project called the Mode ENCODE project for the C. elegans and the Drosophila. And a similar project called the Epigenomics Roadmap project. And the goal is looking for the epigenetic markers, but different from ENCODE, which most focus on the cell line studies. The epigenomic roadmap project is on tissue level or stem cell level. The next couple of things included the TCGA, the Cancer Genome Atlas project. So this is really a project trying to understand the tumors, and there are 33 different tumors types were studied. Involves tens of thousands of samples. And in this project, they not only did the DNA sequencing, which trying to identify the somatic and germline mutations, but RNA sequencing, microRNA sequencing, DNA methylation, proteomics, a very different level of profiling for these samples. A similar effort will be in the ICGC project, which is the International Cancer Genome Consortium. Another resource we talk about is Genome AD, which is a genome aggregation database. For this project, and they basically collected the data from many different consortiums, including the 1,000 Genomes or the Alzheimer's disease sequencing project and many others. And basically, when they collect all these information, they also reprocess all the data using uniform pipelines and did joint variant calling across all the samples. So that is a very, very important data source that people can use. And the first phase of that project is called EXACT, which is the Exome Aggregation Consortium, and the most focused on the exon sequencing. So now the whole genome sequencing is being brought in as well. CCLE project is the Cancer Cell Line Encyclopedia project. And this is focusing on over 1,000 different cell lines. And they basically measured RNA sequencing for all these cell lines at the basal level without the drug treatment. Okay? And the same cell lines, we also have the drug response data from the Cancer Therapeutics Response Portal with CTRP project, which you have the RNA sequencing for the basal level, and you also know these cells, how they respond to different drugs. That is an important resource for us to understand how to do the precision selection of different drugs based on their transcriptome profiles. Links, a library of integrated network-based cellular signatures. This is very similar to CCLE, but the difference is it's not only having the transcriptome level for the basal level, but also after drug treatment, you also have the gene expression levels. And that will include many, many different combinations. So they cannot afford to do this for all the cell lines. If you think about 1,000 cell lines, 500 treatment, that's 500,000 combinations. And with dosage and in the basal level and after treatment, that is a lot of data. So they have to use a platform called L1000. So basically, that is a much cheaper platform, only measure the RNA level for 1,000 genes. And the goal is to use this 1,000 genes to further infer the other genes. But they want to look for the drug-induced changes in these cells. The GEO and the dbGaP, those are the public data resource for gene expression data and the genetic and phenotypic data. And the last is the NIH Human Microbiome Project. And all this project data is in the public domain. We discussed about the multiplexing concept and the barcode. So basically, this is a 6-base oligo that allows you to put many different samples into one lane of run. So if you use 6-base oligos, that allow you to pull 96 samples. And we also discussed the difference between paired end and the mate pairs. So you can see that the paired end is like the same as the single end, except for it sequences both two ends, while mate pairs, not only the sequencing directionality is different, but the middle insertion size is different. So one is about 200 to 500 bases. The other one is much longer. It can go to 5,000 bases. So these two technologies sometimes are jointly used to look at the structure variations. So what we're going to talk about today is very basically discuss the data analysis workflows. What are the major components being included? And also some of the concepts regarding the sequencing quality evaluations. And we'll focus our lecture on the alignment part. So the data processing workflow. This is a screenshot that we had back in 2010. So you can see it's 8 years ago. And this is very early of Illumina days. And I think the data was generated using GC2 platform, even before they have the HiSeq. But if you look at this, and the cell size, the file size is pretty big. And for this particular one, it's a 4.3 gigabase. So now it's very common for us to have a file that is at a range of 50 giga or even 100 gigabase in terms of the raw data. And so if you open it, so here is the size of the file is 4.3 gigabase. And then if we look at how many rows are in this, it's about 38 million reads or 38 million rows. And then when you look at the data, you get into it, it looks like this. It's a bunch of nonsense that tells you nothing for us. And actually they have their meaning. But this is an important one. These are the sequences they have. At the same time, there will be a bunch of numbers or characters that looks like from another planet, but they actually indicate the quality of the sequences. So we'll get into a very much detail of this in the next lecture about the data formatting part. Okay. So the data analysis for the next generation sequencing include 3 components. And primary, secondary, and tertiary analysis. The primary analysis is really trying to get those ACGTs. In the first lecture, we talk about the different level of platforms. So we have Illumina, Solid, and Nanopore, and everything else. But we also know that for different platforms, their way of detecting signals are very different. Some of them are imaging-based, some of them are semiconductor-based, some of them electrical disturbance-based. And from those signals, bottom line is we want the ACGTs. How do we derive the ACGTs from those signals? Those are called the primary analysis. Normally, we do not have to worry about this at all. This is something that the sequencing manufacturer will solve and normally will be done in the core facility. Our analysis starts from secondary analysis, which include sequence alignment, statistics, consensus calling, and assembly, and so on and so forth. So those are the ones that you do not need to know very detailed about experimental design. You will be able to do it. Okay. If I got a sequencing data, I say this is RNA sequencing data from human, and you can just readily to do the analysis, which is doing the sequence alignment. You don't need to know is this from the cancer patients or this is drug treated or things like that. So this is called a secondary analysis. Normally, this is at the data processing level. Well, the third level analysis is called tertiary analysis. This will be more individual application specific. For DNA sequencing, RNA sequencing, ChIP-seq, and everything else, there are specific pipelines for those. And for this, you really need to know the study design, the conditions of the samples, what are the biological questions you want to focus on. So normally for the different level of analysis, the primary analysis will be done in the sequencing core, and tertiary analysis can be done in the bioinformatics core, and secondary analysis. And tertiary analysis, this needs to be done with a team effort, with an analysis team that in many cases include computer scientists, statisticians, and bioinformaticians, and the most important elements in this will be the biologists who design the experiment. Okay. So when we do the analysis, so you can think of this a very, very simplified way of thinking that you got the sequencer, you got the raw data, and then the first step we're going to do is to do the QC. We need to make sure that everything we do is in the quality is good, and if for the bad ones, whether we can get rid of it. And the second part is to do the sequence alignment. So what sequence alignment is, which we'll get into a lot more detail today, is you got a small fragment that is 100 or 200 bases. And then we got a reference genome that we know what the species genome is. can we find out where this small stretch of DNA sequencing coming from? Okay, so that is called sequence alignment. So there is a lot of theory behind that, and we will go through them in a very high level today. And the second, sometimes that each fragment can be mapped to multiple places in the genome. How do we handle that? Those are some of the considerations. And after that, what we need to do is to do the sequence annotation. So basically, you align this here, and whether this is within certain genes or is in the promoter regions, in enhancer regions, and we need to do those annotations. And then after that, the real statistical analysis can start. Okay, so even before the statisticians can or bioinformaticians can get into it, and there is a lot of steps before that we need to process to make the data more manageable. Okay, so before we get into the theory of the sequence alignment, let me give you something about the sequencing quality. So the first question here is, what does the quality really means? Okay, and so if you ask me, you got an experiment you done in our core, and then you ask me a question, is my data good? I really don't know how to answer your question because there are really many different levels we can look at. I'm giving you a very simplified version, which is on the DNA sequencing. So you can see that for this one, you got the reference genome here, and then you got this sequence aligned in these regions, and then each individual reads here and will be represented by different colors, and those colors indicates the quality of each individual nucleotide. So the first level of the quality we talk about is called the base quality, or base call quality. So what this is, if you got a nucleotide sequence, you say, okay, this is AATTCJ, whatever it is, right? And if I know, you tell me the first letter is A, how confident I am, okay? And by the end of the day, initially it was some of the colors, how confident I am to derive that color into an A. In many cases, we assume it's given. It is not. It's based on the physical calculations, and there is a specific rule that we calculate those colors. So basically, the first quality is for each nucleotide in the sequencing reads, we have a quality. So you can think of if we have 300 base for one read, we will have 300 quality for this read, and then sometimes we have 10 billion of those reads in the one sequencing run. That's a lot of quality to look at. But that's the first quality, which is base quality. The second quality is something called mapping quality. So if we look at a nucleotide sequence, this is 100 base, for example, and then I'm align this base into a part of the genome, how confident I am that mapping is correct. And that is called a mapping quality or alignment quality. And the base quality is for every nucleotide, there is a measurement. Alignment quality is for each individual read, there is a measurement for that. The third level of quality is called consensus quality, which we have so many different reads, and then for each individual genomic location, and there is a call to say here is a homozygous variance, and here is a heterozygous variance. How confident I am that this is a variant. So this is called consensus quality. So given all these three together, that is the evaluation for the DNA sequencing quality. Okay, so if in the exam I ask you some of the quality, I will be more specific, and then you need to first identify what are the quality level that I'm talking about. And the next thing is a very small thing, but we need to know, which is the quality scores. Okay, and this is normally it's being presented as a Fred score, and this is the score is published in 1998. And when they do the first human genome project, and initially developed for human genome project, widely used to characterize the quality of the DNA sequence. The score itself, I mean, those are the DNA sequence spectrums. The quality itself is very simple, actually. It's a simple conversion of Q, which is a quality score, is equal to minus 10 times log 10 as a base of this p-value. And this p is a probability that I made a mistake. Okay? So if I'm saying the base quality, if I say this location is A, and if I have a 1% of a chance that made a mistake, and that p will be 0.01, and then this q value is going to be 20. Okay? So if I have a 1 out of 1,000 chance that I made a mistake, the p is 0.001, the accuracy is 99.9%, and then the q value is 30. So this is this very simple conversion is called a Fred score, and this is I'm pretty sure when you hear about the sequencing talks, they talk about my Q30% is larger than 80%. So what that Q30 means? That means over 80% of their sequence base, that their accuracy is higher than 99.9%, meaning that they will make less than 1 out of 7% chance mistake. Yes?
So the P is based on the sequence quality?
It can based on anything. So for the base quality, there will be a P associated to that. So there is a Q score for that. But actually for the mapping quality and for consensus call quality, and they are using the same conversion. Okay? Any other questions? Okay. And we will use this quite a lot in the future as well. And every single base, this Q value is being documented in some way in your original sequencing data. Okay, so but for all of those, just leave it there. We'll come back to revisit that point in the next lecture. And we will more specific talking about the file formats and how those information are being documented. Okay, so now let's go to the main topic of today, which is the sequence alignment. So what is sequencing alignment? It's a way of arranging sequence of DNA, RNA, or protein, so it doesn't have to be DNA, to identify regions of similarity. And basically, bioinformaticians is using that all the time, trying to help to infer the functional structure of an evolution relationship between the sequences. So if you got two sequences, if you know a lot about one sequence, and if you don't know anything about this sequence, if they are similar to each other, you can infer their functions. But the goal here is really trying to identify the best matching sequences. So there are two types of sequence alignment. The first one is called global alignment. And you can define whatever you want, but this figure will tell you all, right? So you got the two pieces of sequences. You want to align them from head to tail, okay? And then you want to see, trying to match everything as much as possible. So this is more on the global alignment. But the bottom one is called the local alignment, so which means you only want to find a small segmentation within these two sequences that are more similar to each other. Okay? And in the next generation sequencing data analysis, in most of the cases, we will refer our alignment as a local alignment. Because for one thing, that we have one nucleotide that is 100 base long, and the other side is the entire human genome. You cannot align that from head to tail, right? It has to be a small piece in the reference genome. So we will refer our alignment as local alignment. And now let's get in a little bit into the alignment theory. So again, I'm going to skip a lot of things because this is not the course talking about the basic bioinformatics sequence alignment. But the goal here is to analytically determine the best alignment between two sequences. Okay, you got one sequence, you got the other sequence, you want to see whether they overlap, they're similar to each other. But the question here is, what is the best? And this is actually it's defined by a scoring matrix or penalty scheme. Okay, I'm giving you an example of a protein case. Okay, so we know that sequence alignment can be done on the protein level as well. So you can see that each of the 20 amino acids is listed here, and the same 20 amino acid is listed here. On the diagonal matrix elements, those are the ones that X and Y, they are identical, right? So that means I found a perfect match. So if I have a match, and then I will have a credit. Did you realize that every number on this diagonal matrix is a positive number? That means you will give a credit because we see my partner that is happy, right? But for most of the numbers of the diagonal matrix, so you can see there's a negative number. That means if this in one sequence is histone, in another one is a different amino acid, and I will give a penalty. So those penalties differ across different mismatches because we know amino acid, they have their own independent, different amino acids have their own features, like the charge they carry, the size they have, they like water or not. So based on those, you will see that different level of penalties. So some of the mismatches are worse than others. So that is reflected by this matrix. So those are the penalty matrix. But in terms of nucleotide sequence, so the DNA and the RNA normally become much easier. And what the rule we normally give is if there is a match, first of all, we only have 4 letters, A, C, G, T, right? And then if there is a perfect match, we'll give a credit. If it's mismatch, we will not give that credit. And sometimes giving a gap. So sometimes you will give a little bit more penalty in that case. So there is a gap opening, you give a penalty like a 3, and for additional gap extension, you will give 0.1. Of course, this definition is a little bit ad hoc. I'm just giving you one example here. So you can see this is a reference, and this is your sequence. Everything is a match, and those are 1, 2, 3. There are 8 base that are matched, so you get 8 as a credit. But here is there is an opening that being opened. So you get a penalty, which is 3. And then for additional 2 nucleotide addition in this gap, you will get a little bit more penalty. So by the end of the day, these 2 sequences aligned as their similarity score will be 4.8. Okay? But this is only a calculation based on this scheme. You can use other scheme as well. But the point of this slide is before we get into the alignment, we need to know to define the similarity. And these are the matrix that you use for that. So the alignment theory, there is, for the global alignment, there is something called a Needleman-Wunsch algorithm. And then giving that a little bit addition will be changed to Smith-Waterman algorithm, which is to do the sequence alignment on the local alignment. Okay, so this is the slides that show you how that will be done, and this is something using something called a dynamic algorithm. Okay, and I will not attempt to explain that, and it's not very difficult. It should be in any of the 101 bioinformatics course. And but bottom line is you will first calculate this matrix, follow certain rule, and then you do the backtracking and to find out what the sequence alignment is, which I'm not going to go detail. Okay, so if you really need to understand the whole process, I would recommend you to go back to study that. It's pretty easy to figure out. Okay, so now I'm going to go into the fast alignment for the short reads, and when we talk about next generation sequencing technology. So when we talk about the high throughput sequencing, the sequence alignment, there are a couple of things we need to consider. The first one is the nature of this data is the sequence reads are short. And initially it was only 25 base, and then later becomes 36, and become 50, become 75, become longer, but it's still pretty short. And another thing is we really have massive amounts. And we're talking about a few billion of those rays that we need to take care of at the same time. So the cost of this the sequence alignment will include the calculation including two components. One is the speed, which means that you need to finish that in a reasonable amount of time, right? So when we generate a flow cell of a sequence, we get 10 billion nucleotides, we need to making sure that it can be mapped or aligned in a couple of hours to at least, at most a day, right? So, and someone did some calculation in the very early phase of the next generation sequencing back in those days were 100 times less than the data that we have. And if you use the BLAST, which is the normal way to do the sequence alignment, it needs something like 300 years to align one flow cell of it. So it's not really feasible. So the speed is really very important. And the second one is the resource that is required, including the memory. Sometimes you need a pretty big memory for these machines. And another consideration is the alignment quality, whether you allow the gaps or not allow the gaps. And in very early phase of the alignment theories, when people use something called a bow tie, and that is really doesn't allow, it goes very fast, but it doesn't really allow you to have any gap in the middle, meaning the insertion deletions will be missed. And now that for all the algorithms, that problem has been solved. And also another consideration is whether I'm going to consider the sequence quality of because some of the nucleotide I'm saying is A if it's a mismatch, but actually the quality of that A is pretty low, and I will have higher confidence on this particular nucleotide sequence. So those are, in some of the algorithms do consider the quality of the base quality of the nucleotides, others don't. And the overall accuracy is important as well. Okay, so our major challenge is we're going through a couple of billion times of dynamic programming is not practical. So the common strategy and the people you can think of, this is not the way that we really do that, but a lot of people in the computer science field, they will think, oh, I will go to make a hash table. So what that means is you are going to make an index or index the reference. For example, you first recorded, given the human genome, recorded AAAA. They are all in those places. So make a dictionary first. And AAAC in those places. And if you have like a 20 base of this dictionary, and then you can search pretty fast, right? So you just go to your nucleotide, you check your dictionary, and to do the searching. That is called the indexing approach. And the problem for that is making a certain number of indexes feasible, but making it very, very long is not feasible. Like 15 nucleotide is too big. And for none of the computer memory can really host that. So now let's really go through the short alignment strategies. There are two common strategies. The first one is something called, it's still the dictionary based or hash table based. This is something called the seed extension paradigm. And in many cases, the space is allowed. So here is how it works. So you got a reference genome, and then you got the query, which is the sequence you're interested in, right? And if I build a 100 base dictionary is too big. That would be full power 100. That is way too big. How about if I only build like 15 or 13 base? that will be still manageable, right? And the 4 power 10, 4 power 13 is still manageable. And then once I have that, and when I got one particular sequence, I only check this top 13 base, and then I will first align this 13 base, and then now I have maybe a few dozen spots in the genome, and then I will see that further extend this to see which one match the best. In this case, probably this one matched the best. Okay, so that is a very simple strategy. It's called the seed extension paradigm. And you can, so that is the first strategy. The second strategy is to use the suffix-prefix tree scheme. And this is becoming a very much like a computer science related topic. And you can build a suffix array, which we are not going to go detail. But another thing that we cannot skip is Burrows-Wheeler Transformation, and which will go a little bit detail on this. Okay, and coming back to this first strategy, which is hash table based, as I mentioned earlier, it's very straightforward. You just build a kth hash table, k nucleotide hash table, and there will be k power 4 power k this many records, and then you do the seed extension. So I'm not going to go too much detail on that. Actually, some of the algorithm established ones are using this algorithm. So I will spend most of my time today talking about the Burrows-Wheeler transformation. Okay, I know this could be a little bit too much for the students who are more biology oriented, but I'm obligated to talk about this because I need to take care of the informatics students in case after graduation you need to find a job, and this is something that they may ask you in the job interview. So let's get through the very basic theory part of it. And this is a Burrows-Wheeler transformation is developed in 1996. Actually, they are not for sequence alignment. And just some people that in Singapore initially figure out to use this transformation to do the sequencing alignment. And this was actually used for the data compression. And I know that a lot of people that are familiar with a compression algorithm, bzip2, and this is actually the conversion for that. Okay, so there are a couple of interesting features of this transformation, which we'll go through. Don't worry about that. But the point is you have the input and you have an output. Okay, input is your sequence or something you want to compress, and the output is your final product. And between the input and output, all the letters remains the same value. Okay, so you have how many A, and you will still have how many A in your output. And their orders will be changed. And if the original string had several substrings that occur often, like in English, we have the, and some of the words, they happens very, very often. And then after this transformation, they will tend to be together. In that case, that will be very easy for the compression to work on, right? So basically, the compression will say, here is 20 the, and they will just record from here, there are 20 the, THE, and then that will make it your novel or whatever being compressed dramatically. Okay, so it's easier for compression. Okay, so before we get into the utility of it, so let's have appreciation. What is the Burrows-Wheeler transformation? Okay, and so if I will guarantee we'll have something about this in the both quiz and the exam. I will guarantee to have something about that. So just follow me. This is not for your hobby, okay? If I have a sequence which is ACAACG, now I want to do the Burrows-Wheeler transformation. The way I do that is very simple. I add up a $sign towards the end. You can add whatever it is, but dollar is nicer. And then you will put this, copy the exactly same thing here. Okay, next step, you will shift the whole thing towards the left side. See, dollar is here, and then the first A come to the back, okay? Do it again, do it again, do it again, again, and again, okay? So now this is your first conversion, and just lift them out. And then next is to sort this new string, okay, based on their first nucleotide. If first nucleotide tied, and then based on second, if second is still tied, based on the third. So point is sort this into this one, okay? Dollar is smaller than everything else. That makes sense? Do you know how to do it? You don't know how to do it? You don't know how to sort? You know how to sort, right? So basically, you put this here, and then the A will go here, and then C go here. So you make this matrix is pretty much dependent on this. And then you sort this into this form. And then dollar A, C, G. And if the first letter tie, which is A, and then you got AC. If the second letter still tie, you sort based on the third letter, okay? So basically that is sorting, okay? And then this is called the Burrows-Wheeler matrix. And then the last column of this is called the Burrows-Wheeler transformation, okay? So from here, original sequence to the transformation, it becomes GC dollar AAAC. That make sense? Okay? You may not know exactly how to do it yet, but it's not that complicated at this step. Okay? If you think this is too complicated, we have much more complication towards the end. Okay? So this is how to make a Burrows-Wheeler transformation. So once this transformation is built, all else shown here is discarded. You don't have this anymore. So this is the only thing that's being left here. Okay. So in order to do make this transition useful, there are two things that we need to take care of. The first one is this transformation is reversible. So from this to this, we have a strategy. Actually, there is a strategy to convert it back. Okay, so that means that this is a transformation that is reversible. And we need to figure out how this is reversible. And the reason this is kind of special is if you normally do sort, you cannot do unsort, right? If you don't record the previous one, the original record. But this is something that you can reverse it back. And the second question is more fundamental question is why this strange transformation can help us to do sequence alignment, right? For me, this is really just a tool to torture us rather than being useful. But we'll see that how this can be useful. Okay, so let's answer the first question first. How do we unsort this Burrows-Wheeler transformation? So if we already have this last nucleotide and the transformed form, how do we get what is the original form? The way to do that is making this invisible matrix. So you can see that you get this after transformation is GC dollar AAAC. So GC dollar AAAC. Okay? Everything in the middle, in the gray area, is invisible. You don't have it. Okay? But the nice part is you have the first column. Why is that? The first column is being sorted. And you have $1 per dollar here. In this, you have 3A. You put 3A here. And then you have 2C. You put 2C here, G, right? So this is sorted. That is how we initially got it. Okay? So now what we do is we'll use this LF mapping rule. Okay? Don't worry about what that really means. Okay? But the general principle of this is the second A here is corresponding to the second A here. Or the first A here will correspond to the first A here. So that is the ith occurrence of a character in the last column is the same text occurrence as the ith occurrence in the first column. It's very, very difficult to understand by this plain English language, but just think through that. And but what that really tells you is if there is it's a rank number 2 in the last column, it is rank number 2 in the first column as well. Okay, that is the rule. Okay, so now let's start to do this conversion. And now we have this transformed phase, and we want to come back to the original phase. Okay, so you see that the transformed phase is GC $AAAC. Okay, and the first column of this is the same letters, but just being sorted, starting from dollar and AAA and CCG. Everything in the middle is invisible. Okay, you cannot see it. And what you do is you start from the first letter here, and this is G. And then you follow this LF rule. So what that means is this G is only G in the last column, and then you go to the only G in the first column, like this. And then the next letter is C. So this put a C here. Okay? So this is your last letter in the original T, and this is the second to last letter. So now for this C, this is the second C, right? And then you go to the second C in the first column, and then this corresponding to A. So this is the third to the last letter in the original sequence. So now this is the third A in the last column. You go to the third A in the first column, write it here. So and then this is A, and then it's also corresponding to the A. So you put the A here. And this A is the first A in the last column, and then you go to the first A in the first column, this corresponding to C. And then this is the first C in the last column, and then you go to the first C in this column, and now you go to A. So you put the A here, okay? Now you should have a collapse here, but you don't have to. And then you can see that this is the second A towards the last, and then you go to the second A here, and then the next one is a dollar sign. So that is the reversion of it. So using this LF rule, you should be able to reconvert your transformed sequence into the original one. Any questions so far? I want you to understand this, okay? We may have another quiz towards the end of this lecture to do it. So you better understand it. Any questions so far? Let me ask again. Do you get it? Don't worry, we won't have a quiz, but you have to figure it out. I will guarantee you we'll have quiz at some time during the lectures, okay? And I will be out of town next week, so maybe this week. So just to let you know. Okay, so this is basically how to convert this BWT converted sequence back to the original one. Sort of doing the unsort, okay? So now let's see, I'm still not satisfied with, well, I don't want to just get a strange transformation and then can convert it back, right? I somehow want to use that as a tool to do the sequence alignment. So how do we do that? Okay, so you can read this. Okay, I don't care, and I never read this. So let me try to figure out what this means. So now I have this, which is GC $AAAC. You can think of this is my index. This is my reference genome. Okay, my reference genome in this case is short. I have 6 letters. I should have 3 billion letters of this. Okay, now I have 6, just for the demonstration purpose. Now I want to search whether AAC is in my original sequence. So this is my query. In a sense, this is my human genome, and this is a nucleotide sequence that I got from my sequencing experiment. Can I do that? How do I do that? Okay, so the way to do that is just narrow it down. Okay, so you start again from the last letter, AAC, and which is C. Okay, here. So basically, initially you don't know anything, but once you narrow it down to C here, and because it corresponds to the first column. The first column, the nice part of the first column is all the letters, they're together, right? So see, here is $AAACCG. All the letters are together. So when to C, you can therefore narrow it down to here. And then what I'm going to see is for this C, and it's corresponding to A, and look at which of these C corresponding to my next letter. My next letter in this query sequence is A, right? So top is my query, okay? So C, and then my next letter is A. In this case, both this C corresponding to A. I cannot further narrow down. But look at this. This is the second A. This is the third A, okay? So now I'm going to this one. I'm going to the second A and the third A. Got it? And then my next letter is also A. This is in my query. And the first one, I hit a $, which is not in my query. So I rule this out. And the second one is A. This is corresponding to the first A. And then my first A is here. And this tells me that I find the AAC in my original reference. That make sense? Let me do it again, okay? You have this reference genome, it's being indexed, okay, here. And then you don't have anything else. The middle part, everything in the middle is black. You don't see it. Your first column, you can derive it from your last column, right? Because it's the same letters, it's just being sorted, okay? Now you have your query sequence. You start from the last letter, which is C. And then you look at, in this case, C is here, okay? So now I'm looking at the second last letter, which is A here. I'm seeing which C corresponding to A in the next. Unfortunately, they both corresponding to A, so I cannot narrow down here. But this is the second A, and this is the third A. And then I'm going to the second A and the third A. Okay, this is where I need to locate this. And then I look at the next letter, which is A. And then I see, okay, the first one, it's not A anymore. So the first one is not. And then this one, it goes to the first A. Okay? So once I see this, I will know that my query sequence is in my original sequence. What is my original sequence? My original sequence is AC, AAC, G, $. So AAC is here. Okay? So this is the way that you need to find out, you can find out whether your query sequence is in this reference. That make sense? Okay? Let's move on. So basically, this is what I said, right? You go there, you look at C, okay, it's here, and then you go to this is second A, and then it narrowed down to here, and then next is A, so you find it, and then you look at this A, and this one is A. This is the first A, you go here, and then you find it is AAC. So that is the way to do that. Okay, but at this step, it only tells you that whether this AAC, this query sequence is in or not. It doesn't tell you where it is, okay? We still need to find out where it is. But even before that, there is another hoop we need to jump, okay? I'm calculating this pretty simply, right? I'm saying, okay, so this is the C is recognized, easy to recognize, and here is the second A, this is the third A, right? How do I know this is the second A, this is the third A? Well, I can count, right? So this is the first, this is the second, this is the third. But if this one has 3 billion letters, and it will take a while to count it, right? So that is not the most economic way to do the calculation. So in order to do this, what people do is you can think of, we will just count here, we will count here, and this will be too slow. This is a naive way to count which A I am at, right? And the way that people did that is they will bury certain markers here. So they will give you kind of a check to say here at this stage, and this is the 240th A, this is the 225th C, and the G and the T, right? So if you go here, you'll find that this is A, and then you just you need to calculate to the previous check mark, and then you know, oh, there's 2 more A after this, and there's a 242nd A. Okay, so this is simply for computational efficiency. I don't have to add up, calculate everything, how many A's that in front of me. And this will be similar strategy, but this is kind of towards the bottom. Oh. Okay, so that is trying to figure out whether the sequence is in this original reference. So the next thing that we need to go forward to look at is where it is, right? Is that in the first nucleotide of the original reference? So again, that I'm here now, right? So I got this is the first A, and I see the AAC. But where am I here? So the next step is a further walk, okay? So you see that you look at A, and then you see, and then, so this is the first step. This is the first C. You go to the first C, and then this is one more step, and then this C corresponding to A, the second A, you go here, the second A, and the dollar. Okay, so you further walk until the number of steps you hit the dollar. That is where you are. Okay, so basically you do 2 additional steps and then hit offsite, and then it's equal to 2. So our original sequence is AAC, and so this will tell you where it is. So how many additional steps you need to go to hit the dollar sign. That make sense? Okay. So there is another thing that is built in, is how many steps that will hit the dollar sign. If this is in total, there are 6 steps. So that is pretty simple to figure out. But if I have 3 billion letters, and it will take a while to hit the dollar sign, right? And how do we computationally solve this problem? Again, there's 2 ways. The first way is for each individual nucleotide, you just first you walk, okay, and then you know how many steps that you are going to hit the dollar sign. So if I'm coming here, you know there's 6 more steps that will hit dollar sign. C, and I would say 2 steps. Okay, come to here. Okay, here is the dollar sign, and 2 more steps. So you know I'm in the second position. That is okay. And a smarter way to do that is you don't record every single part. Rather, you pick a few of those and record. So this one is G, and it needs 6 more steps. This is A, you need 1 more steps. But here is you walk further to the next recording line. So you have 1 more step to hit the offset 1, and then that means you are at the second position. That make sense? Yeah. So basically, in the previous one, this will be easy, right? If I go here and I say, okay, now AAC, I found it. I'm just look at this row, and we know that going 2 more steps, I will hit the dollar sign, okay? But think about that. I have 3 billion letters in the reference, right? And if I have to have one of these letters for every nucleotide, it's okay. It's not necessary. So instead, what is being done is they will just pick some of the numbers, not all the numbers, doesn't record all the numbers. But in this case, among the 6, only record this one and this one. So for G, we know 6 more steps. A, we know 1 step, and we'll hit the dollar sign. And then when you search to this AAC, and then you know that this is not dollar sign yet, and then you don't have an answer here. You just further walk, and then the next one, you hit this one, you see, oh, this one has an offset being calculated. So you mean 1 more step, you will get to this stage, and this stage have 1 more step to go to the end. So you just add them together, you know there are 2 more steps you can get to the dollar sign. Does that make sense? Okay. So that is the way that they do the calculation. So they can do the search. So coming this together, and so basically you want to search AAC, and then you start from the last letter C, and then the first and second C, and then corresponding to second and third A here, and then this one doesn't work, and then go to here, which is the first A. And it look at the first A, and then this will hit the C, and then the first C, and you go there first C, and then you know there's a go to the offset 1. So you 1 more step, you get to the offset 1. So you know that this is starting. There are 2 more letters before me, and to hit the dollar sign. Okay, that makes sense. So this is Burrows-Wheeler transformation. If you think this is, my explanation is not very good. It's pretty hard to figure it out, to follow my logic. Think about that, how I figured it out. Okay, take me quite a while and to figure out without nobody is explaining that to me. Yeah, but trying to go through that, and it's a very, very smart way to do things. But the question is why we want to do this, and we know that this is again that coming back to the original sequencing alignment theories, right? There are 2 things we need to really be concerned. The first one is the resource being used, which is how much memory you need to record, and the second one is the searching speed, right? So these 2 most important things for the next generation sequencing data analysis. Well, for this particular phase, if you think of how much memory are being needed, and actually this is the only thing. This sequence, this reference sequence, and the BWT converted original reference is the only thing you need, right? That is your indexed genome, although indexed in a strange way, but that is an indexed genome. It's the same size of your reference genome. If your reference genome is human, is 3 billion letters, you only need 3 gig of memory, and you can load the entire human genome in that. If you do the other way, right? If you do this, where was that? If you do this dictionary based, if you do 13 base or 14 base or 16 base, it's a full power that number. Sometimes it can be way beyond that what the memory usage. So that's the first feature. The size of the sequence is very nice. It's proportional to the reference genome size. And the second advantage for this is if you think of the query speed, okay, this is linear to the query size. So how much your sequence is. In this case, I have 3 letters. I will just walk through 3 steps, give or take, and I will be able to identify where I am. rather than some of the exponential type of searching speed. So in this case, this BWT is not only small in size, but also very fast in do the query. And that is the reason that this is being picked as the most popular way of to do the sequence alignment. Okay? So you see that the entire index on DNA sequence is the same as your reference genome. And actually, normally it's 1.65 times of the size of the reference genome, because you have all those markers being recorded and then those checkpoints being recorded. But it's linear proportional to the size of the reference genome. And the searching speed is much, much faster. Any question about that? Okay. I know this can be confusing. It's hard to get the first time you hear about it. And I need to go back to think through it, doing the practice on this. Now coming back to this, that alignment tools, there are many alignment. Most popular ones are using the Burrows-Wheeler Transformation, including bow tie. And this was published in Nature. This is the first one that used this transformation. And it was published in 2009. And the initial version do not allow gaps, but this has been fixed. And this is also important because if another very important tool to do the RNA sequencing alignment, which is top hat, is to use the bow tie as their primary alignment algorithm. So another very important algorithm is called the BWA, Burrows-Wheeler aligner, and this is also published in 2009 and uses this Burrows-Wheeler transformation. And this was also published, but this also allowed the gaps. So between two of these, those are the two most popular algorithms being used for sequence alignment. And so those are for short read sequencers. And there is now there's in recent years there is more advance in the people turned around to try to design the most effective algorithm to align the longer reads, like the sequencing, the nucleotide generated from data like a PacBio or nanopore sequencers. And we can see that they have much longer rates. It can be 50,000 nucleotides or longer. And they also have a significant error rate. For the nanopore, it's about 12%, and for the PacBio, it's 10%. So it's very significant, and that will certainly impact our quality to do the sequence alignment. And we also have not one or two such rates, rather a significant amount. And there is, but if you look at the most popular tools that people are using for this, they were all published pretty early, like 2005, 2002. Those are before we have even the next generation sequencing technology. And those are the algorithms being developed in the Human Genome Project or after that to do the EST alignment on the human genome. But the good news is that they are being constantly updated and now can be used for long reads mapping. And there is also a new algorithm developed for the long read alignment. And these new algorithms, they are actually, if you call them algorithms, probably not appropriate. They are more like a strategy to do the sequence alignment. So I'm just putting this particular one here. You can go back to the reference. But the general idea is you got a very long race. You chop it into the middle one, smaller ones, and you only pick sampling some of the middle part, and then you do the alignment. And then those are the ones that give you sporadic signals across the genome, and then you piece them together. Okay, so I'm not going to go detail about this, and this is the one published last year or 2 years ago in Nature Communication, and has been used pretty widely. Okay, We have another few minutes. I need to talk about the evaluation for the sequence alignment. So if we got a nucleotide, and then we map it to the part of the genome, how do I know this is a good spot or bad spot? Okay, and how many mismatches I need to consider to say this is a legitimate mapping loci? Okay, so some people that when they evaluate alignment, they will use straight Smith-Waterman scores. Those are the dynamic programming Smith-Waterman, how many, what are the final scores when you do the back propagation and things like that. So forget about that. And there is also a Fred scale mapping quality calculation that is being offered, which I will go through in the next slide. And then sometimes people also consider other factors like the number of mismatches, the number of gaps, whether it's unique mapping across multiple. I mean, this is a rate that being mapped in multiple places in the genome or not. Okay, so now let me tell you that the one definition is most popularly used. That is the second one here, which is a Fred scale mapping quality calculation. So you will have an appreciation. I hope you have appreciation how this whole thing is being calculated. Okay, so it's a Fred scale mapping quality calculation, and they're based on posterior probability of correct alignment. So what this really means. Okay, so this is the equation will be in the final exam for multiple times. Probably not, but let me go through what this really means. Okay, so this is your reference genome. Supposedly this has 3 billion letters. Okay, and this is a nucleotide that after we go through the Burrows-Wheeler transformation, for example, and they decided here is the best mapping place. Okay, and now let's evaluate how likely, what's the probability this particular sequence is a correct alignment here. The way they do that is they will first look at the mismatch loci. Okay, in this nucleotide, there are 2 mismatch, position B, and everything else is a perfect match. So now, in this algorithm, we'll be able to use the base quality score. Let's assuming that the base quality score is 20. So what that means is I have 1% of chance that this is a sequencing error. Okay, the sequencers made a mistake. Let's say for B location, the score is 10. That means I have 10% of chance the sequencer made a mistake. So putting this together, so the probability of this is a read that is identical as the reference genome is the same, but my sequencer just made 2 mistakes here is this much, right? So I have 0.1% of the chance that this nucleotide coming from here, but sequencers made 2 mistakes. That makes sense? This sounds like a pretty small number, right? And it doesn't look like this is the correct mapping location. However, if you're comparing this with everything else, okay? So let's say now this is the same nucleotide. You say, okay, it mapped here, actually. It's supposed to coming from here. But the chance is that means my sequencers made a lot of mistakes, more than 2, because equivalently, every single nucleotide here is a mismatch, right? If it's supposed to coming from here, but you assume it comes from here. So every nucleotide will be a mismatch. In that case, you will have to time many, many different numbers together, and that is a pretty small number, okay? It's probably 10 minus 1,000, power 1,000 or something like that. And then you assume this one, this one, this one. So you assume everything else. Basically, you add them up together, okay? But if everything is very, very small, actually this guy stand out. This is the most likely one. Everything is more like a 10 minus 1,000. And only this one is 10 minus 3. And then when you add them up together, this is probably very close to 10 minus 3. And this number will be very close to 1. Okay? This is called the posterior probability. So every other read will have very low p, very, very low. It's almost non-measurable. So if mapping is unique, this number is very much similar to 1, even though that this initial probability is very low. Okay? So your mapping score calculation for this is 1 minus this number, which is the probability you actually made a mistake, and then minus 10 times log base 10 of this. Okay? To do this calculation, actually it's not really feasible, because for every single read, you will have to calculate in every other location what are the probability of that. In reality, what they did is just look at approximation, and which coming from the second best matching and the third best matching, and further doesn't go forward about the score calculation, assuming that everything else is ignorable. So, but basically, you can see that the logic for that, this is called the posterior probability, and so just follow the logic, and then let me know next time if you have a question about it. Okay? This is a lot to try to understand for this particular lecture. So in the next one, we will focus on the data formats. And we'll go to different levels of the files, like a FASTQ, the BAM file, and VCL file, and drill it into it to see what are the information being recorded in each of the file formats.